Today we have a really fun discussion set for us to explore. We have with us Georgi Dinkov, also known as Haidut, right? And you're a fixture in the community surrounding the work of Ray Pete, and you've taken it in your own direction. We also have with us a returning guest to the program, Brad Marshall of Fire in a Bottle, who both of whom have been exploring questions surrounding the obesity epidemic and metabolic dysfunction in general from different angles. Brad, your background's in like genetics, right?
Yeah, I guess molecular biology and genetics from, I have a degree in Cornell, and I worked in the lab, worked in a bunch of Memorial Sloan Kettering Cancer Lab after that, and I never went on to get my PhD, but because I don't fit well in the academic environment, but yeah, now I still, this is what I do now, is I'm continue to study and go down that rabbit hole. Some days I think I'll go back to grad school and do my own studies, but we'll see.
So Brad's been challenging these, kind of received paradigms about what causes obesity. I think we all can agree that PUFA is a big problem here, right? I think that's our starting point. Yeah. Georgi, tell us your background. How did you get interested in these topics about health and nutrition?
Mine was actually inadvertent, almost against my will, so to speak. It was by accident. When I was getting out of college towards late 2002, basically that was just around, or still within the dot-com crash area. And basically at the time, my academic degrees are in computer science and math. And basically at the time, nobody wanted to touch anybody with a degree like mine in terms of employment. So what I did is basically I started looking around campus and found there was an outfit called the National Biomedical Research Foundation. Name doesn't really matter, but basically what they were doing is basically it was a nonprofit that was renting some space from the university. I went to Georgetown here in DC, and they had a lot of money from the National Institutes of Health to build this database of proteins and then do some fancy protein sequence matching algorithms. So basically bioinformatics, right? Kind of like the mixture between biochemistry and computer science. So they hired me as a computer programmer, and I worked there from 2002 until 2005, basically one of maybe 2 or 3 people who built several websites, probably the most high profile of which is called the uniprot.org.
What was the time frame of this?
2000 to 2005.
That's funny. So that timeline matches almost exactly the time that I was at the gene ontology.
Yeah.
And so I was also working for a bioinformatics lab, and we were basically unifying the vocabulary so that all of these other sort of sequence databases would have, if you knew you were talking about a gene or a protein in Drosophila, which is a fruit fly, or a mouse, that you were talking about the same gene, because we had this. No, it's not really an ontology. They say, I don't know, not my
taxonomy. There was different taxonomy, right? Yeah.
Right. Yeah. Exactly. To organize all of these databases that were being built at the time. So we were doing very parallel things back then.
I'm pretty sure probably some of the team, like the 40 or 50 different doctors and biochemists that were actually building this. I mean, they were telling us what to do. They were probably interacting with your group. I just didn't know at the time. Yeah. So anyways, long story short, I mean, it's 3 IT people and then 40 or 50 medical and biochemical people, and you want to fit in, right? I mean, if you go to happy hour, what do you think they discuss? They don't talk about computers. Yeah. Our stuff bores them. Their stuff was boring my colleagues, but I was fascinated by it. So I basically wanted to learn more. And I said, what can I do? And even back then, they said, oh, dude, look, if you're doing it strictly for knowledge, you don't need to go to school. Forget about that. The school is just a diploma. You need to actually it's a license for you to get money from the government.
Right.
And they said, basically, you don't need that. Just, they gave me like a intro book on biochemistry and then one book on physiology. And it basically said, just read these, right? And then listen to our talk. Initially, it would be like a baby. Babies learn language and they become proficient in it without anybody telling them about the rules. All they do is observe how other people use it, right? And over time, something clicks. We don't know what it is, but something clicks and they start using it. And that's what I did. Basically, for 2 years, maybe I was just reading and stumbling. Along with whatever it is that they're doing, they were giving a lot of talks and presentations, both at Georgetown and the National NCBI, National Center for Bioinformatics or Biochemistry Informatics. So I was going back and forth between these two places, listening to their presentations. And then eventually, just like they said, something clicked and I was able to actually understand some of what they were saying. Towards the end of 2005, I was already in graduate school again for computer science. Then basically the contract, they had the grant money was expiring. So we had to part ways. And I said, okay, so what do I do? I want to continue doing I kind of want to do what you guys are doing, but on my own, because I have my own ideas. They said, you're not going to get any money from anybody because you don't have any degrees. But look, if you already kind of know the lingo, so if you want to keep it as a knowledge thing, keep going. We'll help you out as much as we can. And I still maintain contact with these people. So between 2005 and maybe up until 2011, when I actually found Repeat, it was just been reading PubMed reading other medical books, right? I mean, that's what they said. So look, once you acquire the language, the rest is reading about other people's experiments and ideally doing your own experiments, but you're not going to do that because you don't have the degrees.
Right. So joining Repeat was like joining Fight Club, huh? It was like this underground, huh?
Yep. Yeah. Basically, it was much. What do they say? Like the Alice in Wonderlands. There's no bottom. You think you've reached the level, and that's the fundamental level. And there really isn't. I think that applies to reality as well. If you talk to a quantum physicist, they'll probably tell you the same thing. There are no fundamental building blocks of reality. At any level, you're going to find a deeper one.
What did your former colleagues think when they saw you getting interested in those ideas from Dr. Pete?
Most of them basically refused to discuss them because many of them were coming from the genetic side, or at least were working on projects that were heavily funded, towards like silencing or activating a specific gene. That was like back in like early 2000s. Now it's about editing, like removing and or adding a specific gene, right? The CRISPR techniques have gotten so much better. But maybe 1 or 2 out of 50 are metabolically friendly. The vast majority of them are basically saying, look, you're born into this world with a specific lot and it's genetically encoded. Later on when epigenetics kind of came into the limelight, I kept sending these studies where basically they said, like, when you expose a pregnant mouse, that's basically the fetus is already formed, to cat urine, right? And the mouse knows that this is a predator. The mouse's offspring is afraid of cat urine and knows that this is a predator without ever seeing a cat, but ever being exposed to any cat. How do you explain that? Oh, it's just a fluke. It's a statistical anomaly. This study will never be replicated. Well, it kept getting replicated. And basically at this point, I think the vast majority of them have either found tenure positions or they kind of, many of them ignore me, but the ones that still speak to me are basically saying, well, what can I do? I was wrong at the time. But I'm working on these specific ideas. I've been funded for the next 20 years. I'm not going to drop everything that I have right now and throw my money away and suddenly start doing what Georgi is doing, or actually what the metabolic theory is saying we should be doing, just because it seems to be better backed by evidence. So we clash a little bit on those topics because I say, well, so what are you actually doing? You're wasting people's money. Well, Georgi, we're not entirely wasting people's money. We're producing valuable research that you may not like the results of our paper, but maybe the materials and methods describe something important that you can glean information from our research, even though you don't necessarily agree with the conclusions. I think Brad kind of mentioned some of that stuff about the papers that he was quoting about the NNT enzyme. Several of the papers kind of indirectly tell you that reactive oxygen species are good for metabolism without actually having that as a conclusion. So I think that's what these people are kind of telling me. So look, we're not entirely useless. And I said, well, that's excellent. For people who get millions of dollars a year, the criteria basically for success is we're not entirely useless.
Yeah. And, it's in some ways, that's one of the things that I do like about the kind of the university system, the publicly funded system, is the idea that people are allowed to do research that isn't necessarily, focused on a specific commercial end goal. So there's a lot of things about the system that I like that it's designed that way, because otherwise, you wouldn't have people doing this. There's a lot of research out there that's done that doesn't seem important, but then later, people go like, oh, well, that was actually really interesting. And you pull it up later and you're like, whoa, and you see the connections. But yes, it is also frustrating to be in a kind of, a space where you're no one wants to think about
It's a very long-term space, right?
It,
It's a very long-term space for 10 years. And basically these ideas are formed at the beginning. And just the way the grant process works, I don't know if you participated that I've written a bunch of proposals for grants.
Right.
You can't really midway say, hey, I was wrong. Okay. I mean, like,
there's no,
I'm going to change 180 degrees, or like even like a 90-degree change. They will freak out. They'll say, what the hell? What am I going to tell my ethics review committee, that we spend all this money for nothing? You just don't do that, right?
Yeah.
So the good thing is, I agree with you, is that basically you allow people, maybe actually when I was young, you allow young people, give them some funding to actually start tinkering with stuff without being always pressed with deadlines and producing whatever the defined deliverables are. And that sometimes, actually more often than not, the real new research and new knowledge actually comes out of such tinkering, right? Without the university system, without some of that funding that basically may be wasted, but you never know, right? We will never get new knowledge. Basically, if you're stuck in the corporate world where everything is tied to a metric and it has to deliver money, right? It has to produce a specific amount of profit, or the investors will kick you out. It becomes perverted, because if you define a metric, if I'm a resourceful person, I will find a way to meet that metric. Whether that's a good result or not is a whole separate story. And I think we should not be going into that direction when it comes to research, because we've seen what happens when corporations take over academia. I mean, recently I posted something on my blog about the Alzheimer. They call it the Alzheimer cabal. Basically, when big pharma is so invested in the beta amyloid hypothesis that basically the majority of the big research centers on Alzheimer's, the academic ones, are getting most of their money from big pharma, and big pharma has all these drugs in the pipeline that are all targeting beta amyloid. Everybody kind of knows that it's the wrong hypothesis, but you don't go to your sponsor and tell them, go pound sand, we're going to work on something else. You say, give me the money and I'll figure something out. You know, a good researcher will be like, yes, we'll work on the beta amyloid, but they'll take 10% of the money and maybe fund something that really works, right? And later on, if you get a successful result, big pharma owns it anyways. So you go back to them and say, look, it's not exactly what you wanted, but it's still a good result. You own it, you know, as intellectual property. Figure out a way to sell it. Maybe you spin a nice story. You say, hey, we were kind of on the right track, not entirely. That's how science works, right? But eventually it's a good result. Nobody's going to shoot you for curing Alzheimer's, even though it didn't exactly cure it the way you promised you did, right? So that's the kind of situation we're in right now. I hope corporations don't entirely take over academia, because to me, that will be the end of really knowledge discovery. Everything will be corporatized. And not that there's anything wrong with seeking profit. When it comes to knowledge, you cannot really put deadlines to an R&D process.
So Brad, where did you just for people just tuning in for the first time about your work, why did you get interested in solving obesity, looking at metabolic pathways rather than just looking for a obesity gene or something, since your background was in genetics?
Right. Yeah. Well, I mean, you know, I got interested in obesity because I was fat. That part is pretty clear, or easy to explain, I think. And so, you know, and so I've struggled with my weight my whole life. And when I was younger, like in my 20s, you know, I did keto, and that worked pretty well for me. It worked less well later. And, you know, really I started, you know, I love learning and reading and researching and reading PubMed. And I was sort of looking around the web for interesting blogs or people with interesting writings and interesting perspective. I stumbled across Peter Dobromilsky, who sort of guided me down the rabbit hole of mitochondrial science and ROS production, and a lot of how that affects insulin signaling within the cell, et cetera. And then, you know, once I had kind of picked that up, it became this very intriguing story that I sort of couldn't put down. And so I'm still, you know, fleshing out that story, but that kind of, and that got me really you know, I'd taken biochemistry at Cornell, right? And you learn all of the pathways. You learn the TCA cycle, and you learn, you know, de novo lipogenesis pathway, and you learn beta oxidation, right? And you memorize it all, and then you take the test, and then you forget it. That's how it works. And it was very interesting. But when you take biochemistry, all that stuff is taught in kind of like, well, here's how it works. Somebody figured out all these reactions, and this is how metabolism works, and it's kind of like end of story. And then they move on and they talk about whatever other things, I don't know. And so, but there's no like, they don't tell you that like, oh, actually, if certain things happen in this pathway, it can shift the whole redox balance of the cell, and like, what does that mean? And so those are the things left out of when you learn biochemistry. And so that's really where I've been approaching things, and why, and the role of ROS is very interesting, because it's right. I'm pro reactive oxygen species, which is, I guess that's a very controversial position to take. I mean, I remember in college I took oncology, and they basically told me that ROS caused cancer, and now I don't believe that anymore. You don't want a buildup of ROS to the point of outright oxidative stress, but the production of ROS, it seems like, is at the very core of how our metabolism works, I now believe. So anyway, that's kind of the road that I went down.
And if I can add a little bit, for most people, and actually most doctors, they call something oxidative stress, when in reality majority of people are in reductive stress, as exactly. You've mentioned in several of your articles. It's most people are under consuming oxygen and under producing reactive oxygen species, and on top of that, many of them are loading themselves up with antioxidants, which further decrease their resting metabolic rate. And you basically, just as you explain in your articles, there's something that needs to happen with these extra electrons, right? And oncology, unfortunately, has taken the view that unless you have a genetic mutation, this cancerization floodgate is not going to open. But now we know more. Actually, in fact, it looks like the evidence has been there all along, but it's been sufficiently either ignored or suppressed. And actually, it's just a simple qualitative change eventually leads to a quantitative one. And if this state of reductive stress continues for too long, you will get de novo cancer. It's not that difficult to actually start it in the lab and demonstrate it happening.
Yeah, this discussion kind of is a spinoff of, we did an obesity panel where we had a lot of the big voices in public who've talked about PUFAs and seed oils. And we asked each one to give their explanation. And we had Dr. Ray Peed and Brad Marshall and Tucker Goodrich and Kate Shanahan and Peter Dobromilsky. We had, and we asked every one of them to give us their best explanation for how seed oils are contributing to this obesity epidemic. And Dr. Pete talked about it being connected to this reductive stress. Yep. And I remember Brad's face lit up. He was so excited when he heard that someone else shared that opinion with him, right? So can you explain for the audience, both of you, how this reductive stress is caused by obesity, is causing the obesity problem, and how seed oils are doing this? Or is it more complicated than just PUFA?
I mean, I'll let Brad, if he wants to speak to reductive stress, I'll try to touch more upon the fact that PUFA is also pro-inflammatory and also has endocrine signaling as well, which contributes directly to the obesity as well.
Sure. Yeah. And so, the topics that we were just talking about are essentially, so what's happening is that due to the way that the mitochondrial electron transport chain is configured, what happens is when you consume PUFA, when you burn it in the mitochondria as fuel, it doesn't burn as much oxygen. It doesn't create as much reactive oxygen species. So it doesn't burn enough oxygen. And that's what you were just saying, is that right, with PUFA, I think you said people are burning too little oxygen.
Yeah.
And I think that is at the heart of what happens with PUFA. And so what happens is, as you're burning polyunsaturated fat in the mitochondria, if you're not generating enough oxygen, you're not able to completely oxidize the fats. And so what happens is you start to have a buildup of kind of metabolic byproducts or intermediates of burning fat, and that often is too much NADH, which is so NAD is a very important, it's an electron carrying molecule, really. It's taking electrons from our food and it's putting it into the electron transport chain, and that's how we make our ATP to move our muscles, etc. And the NADH is the reduced form, and that just means the opposite of oxidized. So reduced means it has the electrons, and so so the electrons come from the food. They make too much NADH, and when you get a buildup of NADH and too little NAD plus, which is the oxidized version, all of a sudden the electrons have nowhere to go, and they start to build up. And the same thing happens with acetyl-CoA. So all of our food gets broken down to this little 2-carbon molecule called acetyl-CoA. And that's what's actually burned in the TCA cycle, which is also known as the citric acid cycle, which is also known as the Krebs cycle. And so that Krebs cycle is really at the heart of how we strip the electrons from the food that we eat. And those electrons are where our whole energy of our metabolism comes from. But if you build up too much of that acetyl-CoA, which is the final step before it becomes completely oxidized, and too much of that NADH, what you essentially have is you have too much fuel in the mitochondria, and there's if you're burning PUFA, you're not generating enough oxygen, and it just simply can't burn completely. And when you have too much acetyl-CoA and too much NADH, that's sort of a switch for the mitochondria, and it does a couple of things. One is it blocks the ability of pyruvate dehydrogenase, which is the enzyme that allows us to burn carbohydrates as fuel. So if the cell is under reductive stress, which means too much acetyl-CoA and too much NADH, you no longer can efficiently burn carbohydrates. And then the second thing that happens is, well, there's really three things. The mitochondrial enzymes that are that actually do this metabolism and generate energy from our food, they actually become acetylated. And you'll notice that acetylated sounds like acetyl-CoA, and the reason is that the fuel itself, when it builds up, you have an oversupply of fuel and not enough oxygen, that actually shuts down the mitochondrial enzymes. So now we can't burn fat efficiently, or carbohydrate, or anything else for that matter, once those enzymes become acetylated. And then the final step is that the lipogenic transcription factors, so that lipogenic means fat making, those lipogenic enzymes are actually activated by acetylation, which is to say that they're activated by a buildup of acetyl-CoA. And so quite naturally, and that is I think that's the correct response of the body. It's like, well, we have a buildup of fuel in the mitochondria, we need to get rid of some of it. And so it gets built into fat, and then it gets exported, and then it gets stored somewhere. And so that actual buildup of energy in the mitochondria is reductive stress. It's caused by insufficient oxygen to completely oxidize the fuel. And it basically turns off mitochondrial enzyme, and it turns on de novo lipogenesis, which is making fat. That's the short version, I guess.
And I would add to that, basically, the response of most doctors to it would be like, you're just eating too much. Well, it turns out actually the macronutrients and their structure and the composition matter tremendously. For example, if your excessive electrons is coming from basically eating too much carbohydrates, de novo lipogenesis from carbohydrates does not kick in until you eat more than a pound a day. And for most people, that's not happening. Most of the extra carbons are coming from saturated fat, from fully saturated fat. People don't get fat either, at least based on the animal studies that we have seen. However, you can actually eat a slight caloric deficit based on your, body mass or body mass index. But if the majority of the calories are coming from polyunsaturated fats, and even to a degree, even the monounsaturated, but polyunsaturated fats are particularly good at doing that, you will actually gain weight because your metabolic rate will drop. And that's something that medicine is just now starting to open its eyes to, because up until now, the mantra has been a calorie is a calorie is a calorie, right? Calories in, calories out. Well, it turns out that it matters what kind of calories you're consuming. And of course, that sounds like somebody from the alternative health will say, yeah, of course, let's all eat salad, right? Let's eat these like non-caloric dense foods. That's not what we're saying here. We're saying that at a very biochemical level, you need to be aware of the composition of the actual chemical structure of the things that you're ingesting. And many of them, specifically the macronutrients, basically, the thing that you probably should be most careful about is the composition of your fats. Ideally, you should be eating as close to fully saturated fat as possible as part of your daily caloric fat intake. Maybe a little bit of monounsaturated fat, simply because purely saturated fat tends to be very rare in nature. And then basically try to do what you can to avoid the polyunsaturated fats, because in nature they're universally associated with torpor, with actually hibernation. And a recent study demonstrated, I've actually had this argument with several doctors who said, we're not squirrels, we're not bears. You can't just take a study that basically looks at the squirrel that's eating nuts in preparing for hibernation, and that eating the PUFA, it's lowering its core temperature, and eventually basically turns off succinate dehydrogenase, as you mentioned in one of your articles, and the squirrel basically collapses semi-dead, right, for several months. That you can't do that. Well, a recent study demonstrated that the PUFA has almost the exact same effect in humans. Yeah, we may not hibernate, but actually that's never been tried. I don't think any ethics committee will allow it to happen. But actually, the core temperature drops significantly when there's an overconsumption of PUFA. Brain function slows down, reproductive function quickly turns off, right? All of these are signs that actually all of these are things that are happening in the hibernating animals as they're preparing to hibernate. So it turns out that we're not that different, at least when it comes to the effects of PUFA on our body from hibernating animals.
Someone on Twitter, or perhaps in the comments of my blog or somewhere, posted a reference to Eskimos from like the late, I don't remember the time frame, late 1700s. It was a very old reference. I have not been able to track down the references. Completely unsubstantiated, just somebody told me. But they said that there was a firsthand report of Eskimos, and it said that they spent their winter basically in a torpor-like state, in like curled up in their like freezing cold tents.
Yep. And they're eating blubber, mostly blubber, basically.
A lot of PUFA.
Yeah. And why is I mean, the people say, well, how do you know it's PUFA? Well, most of the cold living animals have highly unsaturated fats, because otherwise, if they're saturated, they will basically become solid, and the animal is not going to be able to move. So whether it's a seal or a whale or some kind of a fish that they caught there, basically the fat of the blubber, and that's mostly what it is, because that's where the calories are. It's mostly composed of polyunsaturated fatty acids, specifically linoleic and linolenic.
So is PUFA an environmental signal? Has our bodies adapted it as an environmental signal to tell us winter is coming, or is here?
Exactly. Not just winter, but more like starvation, or like in general, inhospitable environment, that basically it's not the time to run around and dance. It's not the time to make babies. Basically, you should just curl up and try to survive. And if you do, then basically you wake up, you reactivate, you deacetylate your electron transport chain, you start eating more proper food, and then all the nicer things in life may happen. But until that time, until that happens, you're basically in a semi-zombie state, and that's how nature prevents you from dying at the cost of other things, such as living a fulfilled life as a human being. So when you're eating PUFA, you're basically evolutionally, at a functional level, you're reverting back to I don't know, I guess a more primitive animal, such as an amoeba or bacteria, which have much lower metabolic rate, and they produce energy primarily through primitive mechanisms such as glycolysis. If you want me to, I can expand now the effects of so the polyunsaturated fats. Now they also have endocrine effects which contribute to the reductive stress. For example, that old study that I mentioned just before we started recording, demonstrating that the saturatedness of a molecule basically can determine its so-called estrogenicity or anti-estrogenicity. And many people think of estrogen as a female hormone. It is not. Actually, estrogen's primary role is as a stress hormone and as a growth hormone. The primary effects, primary physical effects of estrogen on the cell are actually to increase its affinity for water. Estrogen contains the A ring of estrogen is a phenol ring. It's got 3 double bonds, and basically estrogen also has 2 hydroxyl groups. So in general, as a molecule, estrogen is much more hydrophilic than something compared to something such as progesterone, which has 2 ketones and only 1 double bond, or something such as pregnenolone or even dihydrotestosterone, which is the strongest androgens that males make. It's basically has no double bonds, it has 1 ketone and 1 hydroxyl group. And the amount of estrogens that you have in your cell determine how much water basically the cell is going to absorb from the extracellular space. It turns out that the polyunsaturated fats mimic those effects of estrogen quite well. And what happens is when the cell absorbs water, it swells with water from the extracellular space, it actually kicks in it activates some of those same processes of turning off the electron transport chain by acetylation, just as you mentioned, and basically prepares for division and for growth. Starts to reverse the electron transport chain in the opposite direction, and it starts to produce many of these intermediates that are necessary for creating a new cell. So it's an anabolic process, but basically, when it gets out of control, you're essentially getting cancer. Now, for example, I'll give you an example of a good situation when estrogen needs to be high. When you have a wound, right? That wound needs to close, and in order for that wound to close, you need to rapidly create new tissue in that gash where it's there, right? Well, actually, that healing wound, that process is indistinguishable from cancer. If you measure the redox state of the cells that are part of the actual immediate wound, you will notice that they are in a heavily reductive stress. They have very low NAD plus to NADH ratio, mitochondrial specifically, and they have a very high levels of lactate. Why high levels of lactate? Well, when you have a buildup of NADH and there is nothing available to oxidize it, which is what's happening in the rapidly dividing and growing cells. They're not exactly thinking or concerned with differentiation. They're just trying to fill up that gap, right? Then basically the cell can die if it doesn't have a sufficient amount of NAD+. It needs that even for glycolysis. So in order for even glycolysis to proceed, which is the very basis, the very primitive mode of metabolism, you need some NAD+. Well, if all you have is NADH, and there's nothing else to oxidize it, oxygen is not being used properly because the electron transport chain and the Krebs cycle are being turned off, then the only other thing available is pyruvate, which is the output of glycolysis. And pyruvate becomes the emergency oxidant, just like NAD plus is an unoxidant. And pyruvate oxidizes NADH back into NAD plus, but in the process it becomes lactate. And that's the ultimate of reductive stress that you can get. And you can see it obviously in people with cancer, but you can also see it in people with uncontrolled diabetes, especially people in diabetic ketoacidosis. You can also see it in critically ill people that are in the hospital with severe burns, some kind of an infectious disease. Actually, recently, speaking of, since COVID-19 is still a hot topic, they discovered that 60% of the people who basically died in the hospital, 60% of them were already presenting with type B lactic acidosis, which means by definition heavy reductive stress. Wow. At the time they were admitted into the hospital. So the redox balance of the cell matters tremendously, and the polyunsaturated fats, just by the virtue of their structure, even without even talking about metabolism, they're already priming the cell towards turning off its metabolism and basically dividing and growing, basically trying to keep you from dying at the expense of potentially uncontrolled, unchecked growth, which ultimately may also kill you.
Right, and just to add another note, what Georgi was saying about lactate is, so one of the things cells can do is if any given tissue is in reductive stress, you can do just what he suggested, which cells can actually pull in circulating pyruvate out of the bloodstream, and they can reduce it to lactate, and they can re-release it back into the bloodstream. And what that does is it allows them to oxidize a molecule of NADH back to NAD+. So it gives them an NAD+ back. And if any one tissue is out of redox balance, it can do that. And that is the way that the organism can equilibrate redox balance across tissues, right? So if any one specific tissue is in reductive stress, it can use this cycle of grabbing pyruvate, converting it to lactate, and then exporting the lactate to get back to balance. But once all of the body tissues are in reductive stress and they're all trying to use this same trick, where do the electrons go then? And that becomes the problem.
Yeah, and some animal studies have shown that if you administer a very strong oxidizing agent that can oxidize lactate back into pyruvate, such as 1,4-benzoquinone or even 1,2-benzoquinone, also known as ortho-benzoquinone, you can actually save an animal from a lethal lactic acidosis. Lactic acidosis is a 40% mortality rate, even with treatment. And the majority, the current treatment is you just pump people full of baking soda, right? You're trying to equilibrate, the pH in the blood. However, that does not necessarily take care of some of the lactate that is still inside tissues. And also, if the specific organ or tissue is struggling metabolically, it's going to continue to produce it. So something is blocking one or more of the pathways that are shuttling electrons from food to oxygen, and you need to find out what that is and where exactly which pathway that is. And usually, administering an electron withdrawing agent may correct the problem, not always, but it's a step in the right direction. And by the way, PUFA wreaks havoc at every step of this process. It wreaks havoc at pyruvate dehydrogenase, right? It wreaks havoc at succinate dehydrogenase, and it wreaks havoc at complex 4, which is the cytochrome C oxidase. So the three critical steps, at least three, there may be more, PUFA actually inhibits the flow of these electrons at every one of those three critical steps. So it's hard to get a more potent metabolic toxin that is so ubiquitous in our environment than PUFA. Now, there are things that may kill them more quickly, such as carbon monoxide, but its actual effects are very similar to PUFA, just much more immediate. It will bind with cytochrome C oxidase, and you will not be able to basically utilize oxygen, period, and you die almost on the spot. PUFA does the same thing, but in a much more prolonged fashion. So it'll take 10 or 20 years, but essentially the outcome is the same.
Georgi, while we've got here, I want to ask you something that I believe Dr. Pete said during our last conversation with David. He said that if you look at the one of the alternate definitions of acid bases, which I believe is the Lewis definition of acid bases, he said that carbon dioxide itself is an electron acceptor, or carries electrons in the blood. And so so at the heart of what we're talking about here, of course, with reductive stress, is basically what we mean is there's too many electrons. The electrons have to go somewhere. They need to go back to oxygen, is typically how it works. But I was curious about that, and it's not something that I know. I woke up this morning and I started reading the Lewis definition of acids and bases. That was what was in my head upon waking. And so is that something that you know much about, this ability of carbon dioxide in the blood to carry electrons? Because it's very interesting if so, but I don't understand what happens when let's say carbon dioxide is carrying this extra electron pair. What happens when it gets to the lungs? Is it it
doesn't actually carry directly electrons. I asked that specifically because I looked at the structure of carbon dioxide, and I'm not aware of what happens to carbon dioxide if one of the carbonyl groups accepts a hydrogen, right? I'm not aware of such a chemical compound. It doesn't exist. So but carbon dioxide, if you look into the list of Lewis acids, it is listed there. It is acidic. So what carbon dioxide does is the structure of the molecule, because of these extra carbonyl groups, it has affinity for electrons, and its presence in the cell stimulates the flow of electrons in a particular direction. So conversely, something such as, let's say, like a reduced glutathione, right? Or something with a sulfhydryl group. You'll have a repelling effect on the electrons, right? And it will tend to block their flow, or at least be for the electrons to find a way around that cluster of molecules that is heavily reduced.
Right.
So I asked Dr. P directly. I said, "Is carbon dioxide an electron withdrawing agent such as coenzyme Q10 or vitamin K or methylene blue?" He said no. He said, however, its presence there, because of the small size of the molecule and the two carbonyl groups for only one carbon atom, stimulates physically, basically pushes the electrons towards. I mean, it makes it so that there's no stagnation in the flow of electrons.
Right. It basically stimulates electron flow.
Exactly. So think of it as a catalyst.
Something like NADH, which is sort of like a resistor.
Exactly. So
it's lowering resistance in the wire, basically.
Exactly. It's lowering resistance in the wire. That's the because I said, look, I'm not. I mean, if I add carbon dioxide to a solution to basically that has lactic acid, that's not going to change anything. You'll still be lactic acid. He said that's correct. However, you may actually get a phase separation. Like where the carbon dioxide is, you may get like a higher concentration of the lactate atoms because they have an affinity for the carbonyl groups. You're not going to oxidize lactate back into pyruvate. For that, a stronger oxidizing agent may be needed. For example, methylene blue. There's an older study actually with tumor cells which showed that methylene blue is capable partially, not completely, depending on what concentration of methylene blue you enter and how much you stimulate the reaction by increasing temperature and whatnot. But it is capable of directly oxidizing at least some of the lactate back into pyruvate. Carbon dioxide doesn't do that, but just like you said, by lowering the resistance of the wire, it basically stimulates the flow of electrons. Now, if there is something blocking cytochrome C oxidase, such as nitric oxide or carbon monoxide, carbon dioxide will not help. You actually need to release that brake somehow through some other means. But it's still, it's production in the system, because the majority of carbon dioxide is produced in the Krebs cycle, is a testament to a well-functioning metabolism and sufficient also production of reactive oxygen species.
Yeah, that's excellent. It's been the CO2 question, carrying electrons, has been sort of like pinging around in my brain since we talked to Dr. Pete the last time. So that is a very good explanation, and I'm glad you asked him directly, because
yeah, I was confused too. I said, well, it does look like a quinone, right? It's got these two carbonyl groups, right? But I'm not aware of any direct reaction where carbon dioxide gets reduced into something while oxidizing the NADH, right?
Right.
He said it doesn't do that, but without that, actually, he claims that without sufficient amount of carbon dioxide into the system, even adding a quinone may not work very well, because the quinone may actually get processed by, let's say, something like the superoxide anion or one of the hydroxyl radicals. He said the presence of carbon dioxide actually somehow makes the quinone have a higher affinity for NADH than some of the other reactive molecules floating around that can be oxidized. I'm sorry.
Right. Okay.
And the hormonal effects, which I kind of hinted to, PUFA, because it is basically unsaturated. So if it has an endocrine effect similar to estrogen. Now, what's the problem with estrogen? Well, aside from actually making the system, the cell absorb more water, estrogen itself activates the adrenal glands while suppressing the thyroid. Now, the thyroid, if you have a suppressed thyroid, the direct probably doesn't need much of an explanation, but your base metabolic rate, your resting metabolic rate, is actually largely determined by the amount of T3, triiodothyronine, you produce. That is the master controller of your metabolic rate. In addition, basically by activating the adrenals, which produce cortisol, among other hormones, cortisol is actually, whenever you release cortisol, you're also releasing adrenaline. So you're going to increase lipolysis. So in addition to actually having this PUFA from the diet, you're also going to flood your system additionally with additional fat. Most of it is actually PUFA from the fatty tissues. And you're going to have even a more buildup of a reductive stress. And then finally, because many, if not most, of the inflammatory mediators, specifically the prostaglandins, the leukotrienes, and the thromboxanes, they're actually synthesized from arachidonic acid, which is itself synthesized from linoleic acid. Those are highly inflammatory mediators. So the more PUFA you have in your system, the higher your baseline inflammation will be. And whenever inflammation is high, it's just yet another unfavorable signal from the environment. Whenever inflammation is high, cortisol will rise adaptively just to keep it under control. And again, when cortisol is high, it is a signal that things are not going well. So adaptively, the body will turn off most of its other higher processing functions, specifically the electron transport chain, and to a degree, also the Krebs cycle as well. But the key point will be the pyruvate dehydrogenase. There is an inverse correlation between the amounts of cortisol you produce in your blood and the amount of glucose you can actually oxidize. And cortisol, it's the primary regulator of gluconeogenesis. So you're going to be producing even more glucose, even if you don't eat it much from the diet. And you'll be shredding your body, because the glucose will be produced from the amino acids from your tissues, which cortisol will shred. And finally, cortisol also activates the enzyme fatty acid synthase, which you mentioned in one of your articles. Right. So just the whole cascade will be towards not oxidizing the energy that you've ingested, flooding the system with even more energy from actually your own tissues, and then spinning that energy in empty cycles, and then reconverting it back to fat and potentially storing it without actually producing any useful energy, or at least ATP in the process.
Right, and adding to that, kind of line of thinking, when the cell is in reductive stress, there's two key enzymes that are essentially driven by NADH levels. And so what I mean is D6D, which is delta 6 desaturase,
Desaturase,
and SCD1, which is another desaturase. And so there's these two desaturase enzymes. One of them, D6D, is the first step on the pathway of linoleic acid becoming arachidonic acid, which then becomes the oxylipins that Georgie was just talking about, and all of the ensuing inflammation and downstream effects that they have. And so the reductive stress itself is driving the activity of the D6D enzyme, which is what's causing the creation of the arachidonic acid and the other things. And the other thing that the NADH is driving is the desaturase, the desaturase activity of SCD1, which is converting your saturated fat into monounsaturated fat. And this is a subtle thing that happens here. And this is like, you know, people who read my blog and have questions, they like things to be very black and white. And so this is a hard topic for people, because what I always say, you know, the thing I say sometimes is PUFA is the spark, but MUFA is the flame, because what happens in both hibernating animals and obese humans is that the ingestion of PUFA drives the initial reductive stress. But once you're in reductive stress, what happens is the enzyme SCD1 is highly activated, and what that does is it's converting all of your stearic acid into oleic acid. And so when you look at the body tissues of obese people, they're actually full of a lot of monounsaturated fat compared to lean people. That's the end state of all of this. And so, you know, I don't know what your feelings are about.
Same. I mean, if you look at babies, most of their fat is fully saturated, and what they call baby fat, some of the babies probably qualify as obese based on the BMI index. However, if you measure their metabolic rate, is about 50% higher than an adult lean human. And actually, if just as you said, if you actually take a sample from the fatty tissue of a very obese person, it's predominantly composed of PUFA and MUFA. But with the MUFA being about 80%, probably even more of the composition, and the PUFA remaining the other 20 or like 18 to 20%. Many studies have been done like this across multiple ethnicities, and they all show the same thing. So something in your body, even if you eat a lot of saturated fat, once this reductive stress has been established and potentially exacerbated throughout the years by eating a lot of this PUFA, is basically, you either have to eat saturated fats that cannot be desaturated. I don't know if it can. I think palmitic acid cannot be converted to palmitoleic, right? So maybe
SCD1 does convert palmitic acid to palmitoleic acid, but it does it at a much lower rate than it converts stearic to oleic acid. And so what you see is, you know, the end product. So the end product, I'm just going to provide some background here for people, but so the end product of de novo lipogenesis is palmitic acid. So that's a 16 carbon stearic acid, and SCD1 does act on palmitic acid, converting it to palmitoleic, but it's very inefficient. And so, but that actual, they call it the DI16, the desaturase index of 16 carbon linked fats. They compare the, you put, it's palmitoleic divided by palmitic, and it gives you this small number like 0.05 or 0.1. That index is strongly associated with developing type 2 diabetes, and I think heart disease and probably other things as well.
And cancer.
And probably cancer as well, right? So you see that DI16 is the best indicator of SCD1 activity because we don't eat a lot of palmitoleic acid. So you can look at the DI18, and that is the ratio of oleic acid, which is monounsaturated, to stearic acid. And that is less predictive because people eat things like olive oil or other dietary things chicken and pork have a lot of monounsaturated fat too. And but they don't have a lot of palmitoleic acid. And so the DI18 is less predictive of things like diabetes. It's not less relevant really. It's just harder to see it in the blood. But Right. And so that's how the reductive stress kind of plays out, though, is driving these two enzymes forward, and you end up with your body composition being made of MUFA and you're converting a lot of the polyunsaturated fats into the oxylipins and the oxidized PUFA that are part of these signaling molecules, and they're what are activating. The cycle just goes on because then they're activating things like the aryl hydrocarbon receptor and the sterol X receptor, and those go, and those things, once they're activated by oxidized PUFA, they go into the nucleus and they upregulate enzymes such as CD38, such as PARP. And what those enzymes do is they actually basically burn your NAD plus. They convert it.
They
to
consume, yeah.
They consume the NAD plus. So you start out with not enough NAD plus. That stimulates D6D and this oxidation of PUFA. Those oxidized PUFA stimulate these nuclear receptors. The nuclear receptors go out and they turn on these enzymes that that reduce your levels of NAD plus, worsening the cycle of reductive stress. And all this makes sense if, going back to what you said in the beginning, and what I like to talk about in the blog too, is when you look at a hibernating animal whose biological imperative is to hibernate, right? For them, it's hibernate or perish. And so all of these positive feedback loops exist for a biological reason, right? It's not really pathology. And I like to say this on the blog a lot. I don't think of this as pathology. I think of this as how mammals evolved to survive winter and to survive lean times, And so
I guess we're living in a type of winter. We're living in a type of social winter, continuous. That's how we're like that.
Yeah, exactly. Right. And so now we've all put ourselves into continuous winter.
The politicians keep saying we're in a dark winter, so I guess this is what it's like.
They're right, so every once in a while, not for the right reasons, but every once in a while they tell the truth by mistake.
So I want to ask both of you, okay, so if PUFAs and seed oils are causing this obesity and metabolic dysfunction, why is it that so many people can eat high PUFA, high seed oil diets and remain very thin and they just look lean? They can scarf down 3 pizzas smothered in seed oils and everything, and they're just fine. It seems like they're
I wouldn't say the pizza is mostly seed oil. If it's got a lot of cheese, I would say it's a lot of stearic and palmitic acid. I would like the people that I know that are eating all the salads, which are drenched in soybean oil and grapeseed oil and all these kind of things. They tend to be many of them are not lean. They're what they call skinny fat. They have really lean limbs, but actually tend to carry some extra fat in the central region, which is really the worst thing you can be. It's better to have thick limbs and have basically the fat be kind of well distributed across your body than look like the lemon and sticks model, which is in medicine they call it the Cushing syndrome, and it's actually caused by excess cortisol. So, so the people that I know that are really lean, they're actually pretty careful about what they eat. They don't necessarily eat healthily, but they're very peculiar about what they eat. Many of them smoke and drink a lot of coffee, which are metabolic stimulants, both of them. Not very optimal, at least the smoking one. But if you go to a poorer country where people struggle a lot and don't necessarily have good things to eat, you'll tend to get a lot of heavy smokers and heavy coffee drinkers. Go to a third world country. A lot of people, the majority of the people actually smoke and or drink coffee or use some kind of a stimulant.
But we do know Americans that are, lots of them that are eating McDonald's French fries and stuff, and they're staying relatively, they're not
David.
Obese.
So
they're not obese.
So David, this is actually, you asked me this, the same question on the podcast in the past, and so it's something I've been thinking about because I think it's true. I mean, I have a lot, I know plenty of people who eat seed oils and they look great, and so you're like, why? And I think that is where both your genetics and your epigenetics come into play. I think that, like, for instance, so right, because in my mind, yes, I think that the sort of metabolics that we're talking about is uber important. I mean, that at the end of the day is what determines obesity or not, but you have to interpret that in the context of your own genes. And so, for instance, there is again, I'll go back to D6D because it's a good example. And so what you find is that people who have a less active version of the D6D gene. And again, that's the gene that when you're in reductive stress, it oxidizes, it converts linoleic acid to arachidonic acid, which is in the first step of it becoming one of these oxylipins, which is, part of this whole
inflammatory cascade
starting with PUFA, right? And so people with a less active D6D gene are much less likely to develop type 2 diabetes. I mean, that's a study that was just done, and they did this thing called Mendelian randomization, which is, in the absence of being able to do a sort of controlled clinical trial, is one of the best tools that we have for sort of teasing out cause and effect. And Mendelian randomization is looking both at the genes of the people involved in this study, and also the clinical outcomes that they have, and that way they can see, like, well, for people who ate this thing with this genetic background, that happened, etc. It's complicated, but anyway, the point is that so you do see these direct interactions between your genetic background, and so people with a low activity of D6D can probably get away with eating a lot more polyunsaturated fat, because they're not going to kick in that inflammatory cascade, which is upregulating, the aryl hydrocarbon receptor and driving NAD plus levels down. Or, people might have a less active version of, CD38, another enzyme that's taking NAD plus, or they, there's 1,000 different kind of genetic interactions. Another thing I wonder about is, so one of the things about reductive stress is it does, when you have a high NADH to NAD plus ratio, that also drives ROS production in the mitochondria. And so it's possible that depending on your genetic background, things like, are you producing a lot of CD38? Do you have a lot of these stimulated nuclear receptors, which are making these enzymes that are, competing with your NAD plus levels? Is the fact that, okay, if someone eats a lot of vegetable oil, you have this buildup of NADH, and now you're still generating ROS in the mitochondria, and that specific context, that person might actually have a very high metabolic rate, because they're generating these ROS in the mitochondria, but they still have some NAD plus levels left for whatever reason. They might be able to hum along with a pretty reasonable metabolic rate like that for a long time, until something happens, which could be, that they have some, too much, what is it, BPA, right? It's from plastic. It's an environmental toxin that we all get, and it stimulates the aryl hydrocarbon receptor. These other, that one actually, I think
it's a thyroid antagonist. It's actually a thyroid blocker. It's an estrogen agonist. It's an androgen antagonist. It's got pretty much all the bad things that you can think of.
It's all the bad things, right? And so I think there are people that can sort of hum along like that, and they're okay on the PUFA until, sort of the other shoe kind of drops. And so I could see an argument that for a certain percentage of the people, and you actually see this with, I know this is true of the aryl hydrocarbon receptor. And so what you see is the spectrum of some people that if you overstimulate the AHR, you get a wasting syndrome. And that's how it was discovered in the first place, is people that would have this AHR, they would become very, very, they would just waste away to nothing. But then now we know that on the other end of the spectrum, certain people and at certain doses, the people who get this poisoning of dioxins, which trigger the AHR, they become obese. So at different dosages and different levels, the AHR can either make you fat or it can lead to a wasting syndrome. And I wonder if PUFA act in that same manner, where depending on the person, the genetic background, whether or not they've, how much BPA and dioxins they've taken in over their lifetime, if you can end up with this spectrum where the PUFA makes some people lean and others fat,
Well, if you overeat PUFA and you get yourself diabetic to the point of killing your pancreas, which, by the way, is mostly destroyed by the PUFA bioproducts in the bloodstream, type 1 diabetes is a wasting disease.
True.
And they basically die eventually out of cachexia, just like a cancer or an HIV patient, I'm sorry, AIDS patient will do. But to give a counter argument to how come some people can be lean despite eating all this PUFA, well, their number is actually quickly dwindling. If you look at the percentage and the breakdown of obesity versus leanness, at least in the United States, throughout the 20th century, you started with about 6 to 8% overweight. I think less than 1% was fully obese in the United States at the turn of the century. Right. Nowadays, it's 40% are obese. 40%. 30% are basically pre-diabetic, and they're expected to develop at least type 2 diabetes. So whatever that is, even if these people, like the lean people, inherit, so the percentage, and by the way, the highest number, the highest increase in obesity was seen in some of those traditionally lean populations. So even though you may have inherited some good things from your parents, whether that's genes or the expression of those genes, I wouldn't rest on the laurels. It looks like basically eventually, if you screw yourself up metabolically, I mean, no matter what kind of a good baggage you come with genetically or epigenetically, eventually you'll spend all the currency away and end up in the unhealthy group. So yes, some people can do that, but there's an increasingly smaller number of those that can get away with just about anything they want to do. And I'm not seeing that many of them these days. I know of only one. 20 years ago, I knew maybe 5. Now it's only one, and that person is taking stimulants. So it's hard to say what part of the keeping the weight off is actually good health, or what part is.
True. You guys are a little bit, I mean, in a little bit different age cohort than I am. I'm 33, so I guess I'm still seeing the people who are still,
you're seeing young
metabolism
people.
Hasn't broken down completely yet.
Right? 35 is really where you start to disintegrate.
Right.
Yeah, that's true. I guess a lot of my good friends are still in their late 20s and early 30s too. So those are really the people that I'm thinking of, but older people as well. I do know that, that somehow have managed to keep it up, but it's right. Well, I just want to bring up another study on this topic. So there's a study that I like. They took newborn babies, and they took blood out of the cord, the umbilical cord, and they measured the desaturase index of the blood in the umbilical cord. And so, again, the desaturase index is really a indirect measure of reductive stress. And what they showed was that, independently of insulin or blood sugar levels, the desaturase index of the cord blood predicted the BMI of the children, both at the time of birth and still when they looked again when the babies were 2 years old. So if your mother is in reductive stress, that means that at age 2 you'll be obese. That's what that study tells me. And so you see that, the state of reductive stress your mother is in determines whether or not you will be obese. I mean, right. And so you do see a multi-generational effect in humans.
They call it the rule of 1,000. The evolutionary biologists love it, but basically they're saying that's how they explain love. They're saying intense love lasts about 3 years, and they're saying, well, why? Well, apparently it's to keep the parents together for the 9 months of gestation and then the 2 years afterwards, which comes down to about 1,000 days. Right. And what happens in those 1,000 days largely determines the lifelong health of an organism. And most of that effect is actually concentrated while the baby is still in the uterus, is in utero. Right. The first 2 years after that are also important, but they're probably about no more than 30%, and the other 70 are actually what happens to the baby while the baby is still
inside the mom. Does that mean that women, if they really understand what you guys are saying, should stay away from high PUFA chicken and pork and high PUFA foods completely 100% through the whole pregnancy and breastfeeding? Is that really what they should do? Or is that
at the very least during pregnancy, I would stay away from those things, or at least supplement with something that can mitigate the damage. I mean, extra saturated fat, basically supplement some niacinamide, which not only is a precursor to NAD+, but actually inhibits the 2 NAD-consuming enzymes, both PARP1 and CD38. 2 of the most promising drugs on the market right now for cancer that are just about, I think one of them got approved already. One is a PARP inhibitor, the other one is a CD38 inhibitor. And both of them, if you look at the structure, they're nicotinamide, niacinamide analogs. So take niacinamide, it will raise your NAD plus, it will inhibit the consumption of NAD, so it will raise the NAD to the NADH ratio, keep you towards oxidation, more towards oxidation. And this, while in gestation, just being in a more oxidized state, I think it will imprint the baby for life for a much better health.
So
and of course,
even high, but if they don't take those supplements and their interventions, high PUFA meals every now and then can be a problem for the child in the womb.
Well, then in that case, at least keep lipolysis low and make sure you support liver function, because a good portion of the PUFA, when ingested, it can get glucuronidated, and then the mother can actually excrete it. And it will get foamy urine, because the molecules that are formed when the liver attaches glucuronic acid to the fat becomes a little bit like a soap. So that's why sometimes when you wake up in the morning, have you seen like you get foamy urine? That's because you have too much fat in your bloodstream, and the liver actually has taken a lot of that, made it more water soluble, and you peed it out. So when you're eating all this bad food, all this PUFA while you're pregnant, the very least you can do is don't stress yourself, so that there is less fat circulating around in your bloodstream, and not as much gets to the baby, right? And if possible, take something that supports your liver function, so that whatever fat makes it into the bloodstream is quickly deactivated, or at least most of it, by the liver. Speaking about the PUFA, we're not worried about the saturated fats, and not even so much. I mean, MUFA is not ideal, but not nearly as dangerous as the PUFA.
Yeah.
I want to, that leads me to try to land the plane here. What do you do about this obesity problem folks have? Because I came from the space of low carb and keto, and I've interviewed the doctors for carnivore and paleo, and most of them are going to, recommend something like that, but then you've got the approach that you and Dr. Ray Pete and others have been recommending, Georgie, which is, you're drinking, Mexican Coca-Cola and low fat milk and orange juice, which I've been integrating and seeing how that feels for a few weeks now. And then you've got Brad, Brad's the pioneer of the esteemed croissant diet, which was just an experiment of his, but high stearic acid oil and butter in croissants with red wine, right, Brad?
Yeah, I think that will be fine for a pregnant woman, especially because those saturated fats are so important. I think these days the gynecologists, I think they recommend. I mean, they don't recommend, but they're saying, look, if you want to drink 2 glasses of wine every other day, they're not gonna disown you over that.
Yeah, and I mean, on that thread, some of the highest metabolic rate humans in the world that have ever been recorded are essentially humans living on a pure starch diet. And what, specifically what I'm talking about are like, there are studies among rice farmers in Thailand and in Korea, and there was one recently in Thailand, and they did a dietary study, and their diet was like 84% carbohydrate and like 5 to 6% fat, so they're truly eating almost all starch, and those female Thai rice farmers were the highest resting metabolic rate when you adjust for lean mass in any study that I've ever seen. But you also there's Amazon,
the Tsimano tribe, the
Tsimani tribe in Bolivia, they're eating starchy tubers, they're eating things like manioc and yucca, and they also have incredibly high metabolic rates. And so I think that one of the metabolic hallmarks of obese people is low rates of pyruvate dehydrogenase, and therefore they're not good at utilizing starch. They're not good at utilizing blood glucose, and I think that's why keto has such positive effects for a lot of people, because they can't make use of that starch. And so what happens is people, when you eat too much starch, if your pyruvate dehydrogenase isn't working, you're gonna turn on de novo lipogenesis, and you're gonna convert it into fat. But removing the starch and the carbohydrates from the diet didn't fix you, right? It's sort of a, it's like a bypass. You took a bypass, but like the main highway is still blocked, right? It's so to me, I'm thinking more about ways that as a person that I know, I have this kind of like block to using glucose. I'm trying to figure out like, how do I again efficiently be able to burn glucose, because lean people burn glucose. They're good at it. That's one of the key things that kind of defines them. And so it's like, how do I think if you want to get fully unbroken, you want to again get your metabolic flexibility back. And sure, if you want to if you want to fast for a while, or if you go through a time where you're only eating meat and fat, I don't think there's a problem with that. But I think that what you want to be able to do is, switch back to burning glucose when that's what you're eating and when that's appropriate. And so that's like what a lot of I'm thinking about is how do I get out of reductive stress, get pyruvate dehydrogenase working again, and actually be able to burn the glucose that I eat.
Well, back in the day, they were using a drug called DNP, dinitrophenol, which you're probably familiar with it. It's basically a metabolic uncoupler. And right. Since you talked about uncoupling in some of your articles that David sent me, so basically the issue with not being able to burn glucose for diabetic people and for obese people is something called a Randall cycle. So basically, at any point in time, the cell can only oxidize either glucose or participate in beta oxidation. These two pathways are largely what you call blocking of each other. And it turns out that the block is largely at the NAD to the NADH ratio, because the pyruvate dehydrogenase, which is the rate limiting step of oxidizing glucose, it's entirely determined either, well, of course, by the presence of its cofactors, one of which is vitamin B1. Vitamin B1 deficiency is endemic in the population, so that needs to be addressed. But also just the level of whether you are in the oxidative or reductive stress. And burning more fat in general lowers the NAD to the NADH ratio. So you'll be automatically restricting your ability to basically to oxidize glucose. And that is the default state for most obese and or overweight people, because you have extra fat carrying around. Now, when you're not stressed out, when this fat is staying at its fat cells, and then there is not much of a fat floating around in your bloodstream, you will be able to oxidize glucose. In fact, there's a special population, I think it's in the Caribbeans, they're morbidly obese, but they have remarkable insulin sensitivity. They can drink, 8 bottles of Coke in a matter of hour. And then when they measure they call it the area under the curve for insulin, it's basically the same as if 2 hours later is if they didn't drink anything. It's all gone. Right. It's all metabolized. Right. And it turns out that these people have a remarkably low rate of baseline lipolysis, which is the breakdown of fat from the fatty tissue to go into the bloodstream. Why? Well, at this point, it's probably accumulated enough to be either genetic or at very least epigenetic throughout the generations. But it looks like they have a very, very low baseline levels of cortisol and adrenaline, which are the two hormones that determine essentially how active your lipolytic enzymes will be.
So they're living very relaxed time.
Exactly. Yes. And some people say, well, of course, look at how fat they are. Only a fat person can be that fat. I'm sorry, or a relaxed person can be that fat. Actually, extreme relaxation, I would expect to make you lean, but maybe there's something to be said for that. Anyways, long story short, if you're carrying extra fat around, you will have problems oxidizing glucose unless you find ways to lower your lipolysis, which usually means lowering stress, or you're changing.
Lipolysis is fat burning. You don't want a fat burn.
Not fat burning, fat shredding, which then leads to fat burning, or maybe, well, actually not necessarily fat burning. So it's the process of getting fat from your fatty tissue into the bloodstream that then gets carried around to the cells. But if that fat is mostly polyunsaturated fat, just like Brad said in his blog, it will not get burned. It actually gets emptily recycled back into fat through the fatty acid synthesis, right? So you'll go through beta oxidation. Each one of these acetyl-CoA molecules will then go back into the Krebs cycle. But instead of getting oxidized and eventually producing ATP, a lot of it will actually get turned right back into fat through the fatty acid synthesis pathway.
That's why Ray Peets told us that you don't want to burn fat, you want to pee it out, right?
Yeah, exactly. You want to pee it out, for which liver function is very important. But also other things that I've noticed that elite athletes almost never diet. They do hormonal things, right, or some other interventions, but they basically manage to sort of like get rid of this fat without the exertion, right, or without the stress which goes with dieting and or extreme exercise. You can actually get yourself diabetic if you carry extra weight. If your BMI is over 31, let's say 31, and you start running suddenly to actually lose this extra weight, you may get yourself to a situation where lipolysis is so elevated you can actually shut down the pancreas and give yourself type 1 diabetes. It's not a rare occurrence. So you shouldn't, and that's why now doctors, they kind of say, look, take it easy. If you're very obese, or if your BMI is over 31, they're saying start with walking, don't overexert yourself, right? Don't stress yourself too much. You need to get to a point where your cells are not going to get flooded with too much polyunsaturated fats, which not only will get emptily recycled back into fats, but their byproducts, especially the peroxidation byproducts, which is malondialdehyde, which I think is the main one, they're directly carcinogenic and actually can destroy many of your organs just because they're toxic themselves. So you don't want to, if you're carrying extra fat, you will have some problems oxidizing glucose. You can remediate some of that by raising artificially the NAD to the NADH ratio by either taking niacinamide or taking something that can oxidize NADH back to NAD, such as an oxidizing agent, right? Or you can take an uncoupler. Dinitrophenol is banned by the FDA because if you take too much, you raise your temperature so high you can die from hyperthermia. It has happened. Yeah. That doesn't prevent people from using it, but I think the rumor on the street is most of the elite athletes who want to lose weight, they either use DNP or they inject anabolic androgenic steroids. And what do the anabolic androgenic steroids do? The vast majority of them are uncouplers. They also block cortisol, they block adrenaline, and they help your liver process the fats. So if you look at the bodybuilders, I mean, they consume tremendous amount of food, but they convert most of it into muscle and very little of it into fat. Testosterone and some of the other hormones that are known as anabolics, actually, if you look at their action at the receptor level, the vast majority of it is blocking cortisol and estrogen at the receptor level. And those two hormones are metabolic inhibitors. So by blocking them, you can actually get to weight loss. There's a drug on the market which was currently sold as an abortion pill. It's called mifepristone, also known as RU486. It's in the news lately mostly because they're using. I mean, it's if you want to do an abortion, chemical abortion, that they give you this pill, right? But not many people know that this drug was developed as a cortisol blocker. And when the company Sanofi, a French company, when they developed in the 80s, they realized at the time that there isn't much of a market for a cortisol blocker because the Cushing syndrome cases are not that many, right? But their brilliant marketing department realized that this drug also blocks the progesterone receptor and as such induces abortions. So this drug is in the pipeline. I mean, you can get it by prescription, and it's being used off-label to cure even extremely morbid obesity. Go to Google and type mifepristone obesity or RU486 obesity, and you'll immediately get the studies. All of them.
It's also being researched for anti-cancer effects. I remember
exactly for anti-cancer effects as well. So blocking the effects of the anti-metabolic hormones such as cortisol and or estrogen. This is one of the drugs that does that. I'm not saying it so people go and take it. I'm saying it that doing things that block the effects of these stress hormones actually unleash the brake on your natural metabolic rate, right? Which all comes down to the environment. We are obese because both we're eating the bad things and also because the environment is such that it's telling us, you can't afford a high metabolic rate. You need to be semi-hibernating and packing on the pounds, right? Right. So by blocking the effects of cortisol and estrogen, which are the endogenous signals reacting to the environment and rising and saying, look, you need to stay at a low metabolic rate. By blocking these two signals, anti-metabolic ones, you will probably be able to lose a lot of this extra weight that you have, and you get back to the metabolic flexibility, which Brad said, which means you can very quickly and seamlessly switch between oxidizing fats and carbohydrates without much of an effort or without much of a block. Obese people, old people, and sick people have a block, and they seem to be stuck predominantly into oxidizing a little bit of fat and then emptily recycling the rest of it into that like fatty acid synthase cycle. But they cannot oxidize carbs very well.
Georgi, one of the things I've been thinking about recently is, I've been thinking a lot more about insulin because, coming from the background of a ketogenic diet, insulin is thought about as bad because it is, it's telling you to take in blood glucose. And if you're, if if you have, like you say, a lot of circulating free fatty acids, if you're in reductive stress and you take insulin, that can cause you to uptake blood glucose and turn it into fat.
Yeah.
However, the other thing that you've been talking about is how if you have a lot of fat circulating in your blood, that is going to keep you in, or can keep you locked into this reductive stress, because when you're burning fats through beta oxidation, it does decrease the NAD plus to NADH ratio. But, Peter from the blog Hyperlipid would say that the purpose of insulin is to block lipolysis. And so lipolysis, just as background for people, is that is the process where fat cells release free fatty acids into the bloodstream, and the free fatty acids are really what your cells take up to use as fuel to burn in the mitochondria, right? And so insulin actually blocks the process of lipolysis of the fat cells releasing cells, and that sounds bad if you want to burn fat. However, if you want to get out of reductive stress, the idea, right, in the keto world, and I assume a lot of the people listening to the show are in the keto world. The idea is, you're doing a lot of fasting. You're not eating a lot of foods that stimulate insulin, because you want to keep your fat cells doing lipolysis and releasing this fat to burn the fat, essentially. However, I've been thinking more about, these rice-based cultures, and the idea that, okay, but if we're eating multiple meals a day containing carbohydrate and we're stimulating insulin every time you do that, lipolysis is reduced, but that can help get our cells out of the reductive stress while that's happening. And so it's like, if I were to eat, I don't know, 4 or 5 meals a day of rice or something, every time I stimulate insulin, perhaps that's helping to relieve the reductive stress. I don't know, Georgie, what your thoughts on that are.
Right. And in fact, people have lost weight by just eating rice.
Right. And if you could, a question for you is, how do insulin and cortisol interact with each other?
Oh, they're almost perfectly correlated. Basically, like, if you have hyperinsulinemia, chances are your cortisol is high. And basically, the cortisol could be high for many reasons. Could be high baseline inflammation, could be high stress. Another reason not to eat the PUFA, because you eat high PUFA, your baseline cortisol will be high. And because the baseline cortisol is high, because cortisol stimulates gluconeogenesis, you'll have a reactive hyperinsulinemia, because also the body tries to prevent the blood glucose from going too high, right? The same thing. And the same thing happens in the reverse. Basically, if your insulin is basically, I think if insulin is too low, cortisol will also basically be too low as well. So
if I'm an obese person and I suddenly start eating rice 5 times a day, what's going to happen to my cortisol levels? Are they going to rise in parallel to the insulin levels?
So it depends on what's the reason for your high cortisol levels. After you're obese, I think the problem with many of the obese people is because of a lot of the PUFA that they have, and because they have elevated levels of baseline, I emphasize baseline lipolysis, they have also elevated levels of baseline inflammation. So if you look at their CRP, the biomarkers for non-specific inflammation, such as C-reactive protein, erythrocyte sedimentation rate, some of the interleukins, like interleukin 6 specifically, they're all elevated in obese people, right? And doctors are confused, saying, well, why? I mean, why? Well, chances are that if you look at their non-esterified fatty acid profile in the blood, you will see that it's predominantly composed, or at least 40% will be of the actual already preformed arachidonic acid floating around, ready to be converted into the prostaglandins and leukotrienes. Right. And one of the things kind of corroborating that the reason cortisol is high in obese people, that the inflammation is involved, is that if you give these people anti-inflammatory drugs, their cortisol levels decline and their insulin levels also decline. Their insulin sensitivity improves. So there's something going on in those obese people that elevates baseline inflammation. And I see no other factor aside from stress which can cause inflammation through a non-PUFA manner. I think the fact that these people consume a lot of PUFA and have a lot of PUFA in their tissues, that's really the basis for their insulin resistance, because their base inflammation rises, elevates their cortisol, and cortisol actually blocks the insulin at the receptor level. If you administer a cortisol agonist such as dexamethasone, you can make a person temporarily diabetic. It's well known. There's something called the dexamethasone suppression test, where you go for them to test you whether you're producing cortisol because of a tumor or just because of stress, right? And they give you the synthetic glucocorticoid, very powerful. They tell you, but very, very, very often you're going to have elevated blood glucose for several days later until you actually metabolize this glucocorticoid and excrete it. So cortisol blocks the effects of insulin at the receptor level as well. Now, if a person who is already obese starts eating rice, they may have, I don't think that their insulin will go that much higher because they're probably already at the most that
not the baseline insulin.
Right.
Not the baseline insulin. What happens immediately after a meal is obviously insulin rises or is released after a meal. Is the cortisol affected after a meal? Do you know?
Cortisol also tends to rise, and so far, up until like maybe a few years ago, there was no clear explanation as to why. It looks like it's because of the reactive endotoxemia, which occurs almost any kind of a meal, after almost any kind of a meal. So when you're getting this food, right? Many people these days suffer from something called SIBO, small intestine bacterial overgrowth. There's always bacteria in your colon, right? Anytime you get any kind of a food to the bacteria that has not been fully digested, this bacteria will use that food to reproduce and divide and basically have a turnover. And some of the gram-negative bacteria in your intestinal tract contains this thing in its membrane called lipopolysaccharide, also known as endotoxin.
Yeah.
And it releases it when it dies and breaks down. Some of it gets into the bloodstream, and that creates a highly inflammatory reaction, and cortisol rises. So it's been shown to occur, the rise of cortisol has been shown to occur from anything other than pure glucose or something that gets absorbed directly, even entirely in the stomach or the very top of the small intestine. Anything that goes further down, anything that basically gets into the hands of the bacteria will probably trigger some kind of a even small inflammatory reaction and then reactive cortisol increase as well. But for an obese person, if he starts eating or she starts eating only pure, well-cooked white rice, I think over time their metabolic condition will improve because the insulin will keep lipolysis, which is already elevated, right? It should be able to bring it down to more baseline levels, and there's always baseline lipolysis. And then whatever muscle these people have at rest, muscle prefers to oxidize predominantly fat. Heart muscle is a very good example, but also your skeletal muscle, which kind of gives you an idea of what's the best way to stay lean, build muscle, right? It's a bit of a catch-22. How do I build muscle if I'm already obese? But if you already build the muscle, then it'll be more difficult to become obese. Anyways, long story short is that you don't want to have excessive lipolysis because it poisons your pancreas. It blocks also the effects of insulin at the receptor level. The fatty acids have a receptor, and basically when they activate it largely prevents insulin from acting on the glucose transporters GLUT1 through GLUT4, and this prevents the glucose from actually being absorbed into the cell. You may be producing a ton of insulin, but if there is a sufficient amount of fatty acids in the blood, you will not be able to absorb the glucose from the bloodstream. So keeping lipolysis low, there is a drug on the market which is a derivative of niacinamide. The drug is called Acipimox, ACIPIMOX. Its main effect is lowering lipolysis. And that drug, just by taking it, results in decreased blood glucose, even though it actually decreases the fatty acids. It tells you that ultimately it's the fat that's the problem, the reductive stress, right? It decreases the excessive lipolysis, which allows insulin to do its job, which means blood glucose declines, also drops because it's being uptaken by the cell, right? And also it decreases cortisol, which tells you that the reason cortisol is there partially is basically because of the high insulin and the high free fatty acids and the high glucose. So cortisol and insulin always go hand in hand. Cortisol is there to keep your blood glucose from going too low. That's the primary role of cortisol. It's not inflammation. Your brain, you can go into hypoglycemic coma and actually die. Diabetic people know that very well. If your blood glucose levels drop beyond, below a critical level. Cortisol's job is to make sure that never happens. So people with Addison disease who actually cannot produce cortisol are always running around carrying actually glucose pills with themselves and a little bit of cortisol, of course, because they inject themselves just like the diabetic people to prevent their glucose from going too low. So probably the primary role of cortisol is actually glucose homeostasis. In other words, preventing it from going too low. The insulin role is the exact opposite, preventing it from going too high, right? And these two hormones are always released more or less together, with cortisol, of course, having an anti-inflammatory effect as well. But the primary effects on cortisol and insulin that you release, that you see after eating, are related to the amount of fat and glucose you're ingesting, right? And carbohydrates, not necessarily glucose, and eventually gets converted to glucose. And also the levels of baseline lipolysis. If you're eating something that stimulates insulin too much, right, then you're also going to have a breakdown, a rise of cortisol, because if insulin gets stimulated too much, the body says, oh my God, I'm in danger of blood glucose going too low. Let me upregulate cortisol as well, just to make sure this never happens, right?
Right.
Also, if baseline lipolysis is elevated in obese people, diabetic people, stressed people, because the free fatty acids are blocking insulin from doing its job, then you're going to have an elevation of glucose because it's not being processed, it's not being uptaken by the cell. So ideally, what you should be doing is keeping stress low, and if that cannot be done, doing something artificially to at least keep lipolysis levels at the baseline level and not excessively elevated, because that's essentially mimicking the reductive stress, creating the reductive stress. And finally, all these things should be done in parallel ideally, not ingesting as little PUFA as possible, right? These are the three factors: stress, which creates reductive stress directly, right, because of the lipolysis and then the ensuing cascade through the fatty acid and random cycle, right? Number one. Number two, what are you eating, right? Don't eat too much PUFA, because it also starts the exact same cycle. It puts you eventually into reductive stress and obesity, right? And finally, make sure that the hormonal milieu is such that basically you're not favoring cortisol, which is an anti-metabolic hormone. Whether you do that through avoiding stress or blocking cortisol, that's up to you. I mean, ultimately, I prefer to do it the natural way, because otherwise we become dependent on a pill, right? And life will somehow find a way to adjust to this higher level of baseline stress, just because you'll say, oh, I can always take a pill to take care of it. That's not how it should be done.
So, Georgi, does that, I thought Ray P says that, starches are actually more obesogenic than like sugar, drinking the actual sugar.
Mostly because of mostly because starches, because of the small particle size and the way the intestinal wall is structured, they can actually persorb and get into your bloodstream. Anytime you get a foreign particle like that of complex shape into the bloodstream, you're going to trigger some kind of an immune reaction. So starches tend to be more inflammatory if they persorb into the bloodstream. And that tends to happen in a lot of people because they have compromised gut barrier, because they're drinking a lot of alcohol. Stress actually increases the gut permeability, right? So if a young healthy person is eating starch or some kind of or sucrose, right, they're not going to see much of a difference. Maybe a little bit of a difference in the insulin response, because the starch will metabolize predominantly to glucose, right, which tends to have a higher insulinogenic response. But in terms of overall health, a young person does not necessarily distinguish metabolically between sucrose and starch. For an older, more obese, or a person in more compromised health, if you're going to be eating starch, make sure you eat it with a lot of saturated fat, or at least some saturated fat, because the saturated fat not only tends to limit the persorption of starch into the bloodstream, but some of the fatty acids actually tend to act as antagonists directly on the endotoxin receptor TLR4. Stearic acid is one of them. I know Brad seems to like stearic acid, but it's got very unique properties. Very good fatty acid.
Yeah, you went dark on us, Georgi, but
I don't know what's going on here.
Yeah, I'd like to hear once we get the light situation figured out, I'd like to hear your explanation again of what the saturated fats are doing. Just say what you said there again about eating saturated fat with starch, because
it forms, first of all, the long chain saturated fats stimulate the release of bile acids and the formation of something called chylomicrons. So it's going to increase actually the absorption of these saturated fats through the lymphatic system and distribute them around the body. Otherwise, they go mostly through the liver. But the reason you want more of the saturated fat acids into your lymphatic system is because they tend to have an anti-inflammatory effect, and they also are capable, some of them, of blocking directly the endotoxin receptor, which, as I mentioned earlier, is a very pro-inflammatory molecule.
Got it.
It acts on a number of different receptors known as toll-like receptors, TLR, 1 through 9, I think. But TLR4 is the main one for endotoxin, and some bacteria, which tells you that the body treats endotoxin like a bacterial infection. That's why it causes fever and a lot of other symptoms like that, right? Saturated fats are capable, some of them, specifically stearic acid, and I think palmitic to a degree, are capable of blocking that receptor. So even if starch persorbs, right, basically a lot of the inflammatory cascade from eating starch seems to be due to two things. Number one, the immune system recognizing the starch particles as foreign, mounting a response. And two, with the starch, the persorption of the starch, also some endotoxin is also persorbing into the bloodstream. So if you're eating the saturated fat, it tends to limit the amount of starch persorbs, and then if it does, it's going to persorb together with the saturated fats, and in the endotoxin response portion of the inflammatory response to eating starch will tend to be largely blocked by saturated fats. Right. I think there's a few studies showing that if with volunteers that if they eat like forgot it was some kind of a starch, it was a grain meal or a rice meal. In one case was eaten with butter and or coconut oil, and in the other case I think was eaten with soybean oil and just on its own. And when it was eaten with the saturated fats, the coconut oil and the butter, there was almost no endotoxin response post meal.
I remember that. I saw that study too, and I thought it was pretty cool. But one of the things that, just as to what you just said, and again, how it all adds up to reductive stress, is that, another effect of the endotoxin is that it stimulates the CD38 enzyme, which is again degrading your NAD plus, and another thing drawing down. So anytime you have this inflammation, the body's response to it is to make this enzyme, which is lowering NAD plus. And so it all, it's always seems like it's a positive feedback loop to to get rid of the NAD plus.
You can never get enough NAD plus. I guess that's really the message of the show. Keep it as high as possible by avoiding reductive stress, right? Right. One aspect of it is avoid the consumption of PUFA. Stress obviously consumes a lot of NAD simply because the PARP and the CD38, these are all cell repair enzymes. And repair of cell requires energy, of which NAD plus is perhaps the cardinal representative of. Many people think of ATP, but it really isn't. ATP is more of a signaling molecule and a structural molecule. I don't know if you've read the work of Dr. Gilbert Ling. His so-called association induction hypothesis. Well, it looks like the primary role of ATP is to structure the proteins of the cell together with potassium. So it's basically a so-called cardinal adsorbent. It has high affinity for potassium and for proteins, and it keeps the cell structure. So when the cell is low on ATP, it tends to have the proteins inside tend to get disorganized. And eventually the cell may even rupture and spill its guts into the bloodstream. Speaking of which, another reason why stress causes inflammation even in the absence of PUFA. Recent studies show that the presence of mitochondrial fragments into the bloodstream trigger, for unknown reasons, the exact same response as a bacterial or viral infection, which of course involves the fever response and subsequently the elevation of cortisol to keep inflammation down. So something about the body, basically, I guess it's an alarm signal that if the body is breaking down, the rest of the cells are saying, "Oh my God, all hell is breaking loose. I better ramp down my metabolic rate, and I better start getting into reductive stress and building up all of these extra resources that I have in terms of energy, right, fat, in order to be able to deal with the bad times that are coming." So stress directly makes you fat by putting you in reductive stress, at least the mainstream medicine at least recognizes the fact that cortisol is obesogenic hormone. And some doctors are now saying, let's give obese people cortisol blocking drugs such as IO486. There is a clinical study right now in Stanford to actually treat people with obesity with no other disorder, at least that they claim that no other disorder, to treat them and see if they can cure their obesity simply by blocking most of the cortisol at the receptor level.
So I want to wrap things up. This is fascinating, but we're out of time. Just to conclude, Georgie, what is your, like, if you don't have access or don't want to do those types of artificial drug interventions, what is the healthiest way to cure obesity using, dietary means? Is it to go really hard on the sugar or to gradually increase your sugar? I mean, what is the bottom line, best, healthiest way to cure obesity without being, dependent on these artificial drugs?
I would say maintain lipolysis. I mean, try to prevent lipolysis from rising too much and support liver function so that whatever fatty acids are floating around and this extra weight that you're carrying basically can get processed by the liver. Do some forms of concentric exercise that can build up some muscle, because the muscle burns predominantly fat at rest, right? And then as your weight, depending on how obese you are, as your weight gradually comes down, then you can start thinking about other forms of exercise, or potentially maybe like going on a fast for a couple hours or maybe half a day to increase the fat burning if your liver can handle it. Now, if the person is really is carrying some, if they're in the obese category, I would not directly jump on fasting or exhaustive exercise, because if you flood your tissues with sufficient amounts of free fatty acids, most of which are PUFA, you're going to damage a lot of organs. Kidney failure was recently confirmed in diabetic people to actually be due to chronically elevated free fatty acids, PUFA actually, in the blood. Pancreatic failure, the beta cells of the pancreas are directly destroyed by free fatty acids floating around. Again, exclusively PUFA, saturated fatty acids, and oleic acid did not have the destructive effects that PUFA had. What else? Alzheimer's disease has recently been proposed to be classified as diabetes of the brain. Which means, once again, it's probably a disease of excessive productive stress and polyunsaturated fat presence. So avoid stress, support body functions that can take care of this extra fat, these extra electrons, until you get to the point where you actually can afford to maybe push yourself a little more. Ultimately, the goal is obviously to get rid of these extra electrons that are sitting on top of you.
So you believe that fat burning is healthy? Because Dr. Pete seemed to suggest that you just don't want to burn any fat ever.
Depends on the person. I mean, a lean person that carries, 2, 3, 4, 5 extra pounds, they can probably afford, doing like a week or two of slightly less calories, right? Or like slightly more exercise. But if a person is having trouble, is catching their breath, climbing 10 stairs, carrying that much weight, I wouldn't overexert myself. I would work on slowly building up the muscle mass and, working with the liver to excrete a lot of these things.
Would you be doing a low-fat diet, a no-fat diet, or high carb, 5,000 calories?
You can probably do that. Suggesting? Yeah, I don't think there will be a problem. But
super high sugar and moderate starch, moderate protein, and no fat. Is that the best way, you think?
Yeah, because a lot of these people, almost universally, if you have an obese person, they have some form of fatty liver disease already, and that fatty liver disease is actually due to these free fatty acids that are floating around. The liver can only handle so much of them, and the rest that it cannot handle actually it stores within itself in the form of triglycerides. So for the very low fat, high carbohydrates, take it easy on the protein too, because very overweight people tend to have kidney problems too, right? You don't want to overburden the liver with too much the kidneys with too much protein. Maybe like a gram per kilogram of body weight a day would be the max that I would do for an obese person. And then the rest will be carbs, easily digestible carbs. Don't do the carbs that get to your colon and get metabolized there, the resistant starches, because then you're going to get, basically you're giving yourself an endotoxin attack.
And white rice is an easy carb that you would throw in there?
Well-cooked potatoes, white rice, even grains, if they're really well cooked into a mush, to the point where basically when you actually eat it, it feels like like a puree, not like something that you need to chew on. Those kinds of things. And of course, keeping digestion flow going. If you feel like you're getting an endotoxin attack after eating, eating some insoluble fiber. I know Dr. Pete likes the carrot salad. In my experience, charcoal actually works really well. He says he likes it, but he doesn't recommend it because he's concerned of the quality of the products that are on the market. So with that caveat in mind, I think charcoal can be a useful additive because it keeps endotoxin. Actually, it can kill a lot of the bacteria directly. these carbon particles that are part of charcoal, some of them are actually in they're in a structure that can oxidatively destroy bacteria. So taking charcoal has a mild antibiotic effect. Keeping stress low.
One of the things that I like to do with, speaking of grain, is making masa the old-fashioned way. So the Native Americans, they would take their corn and you actually boil it with wood ashes. So you're adding charcoal to the water and you're boiling that with the corn, and it releases the B vitamins in the corn and it changes the texture to make it sticky.
Is that like hominy? Is that like
hominy? That's what hominy, right.
Yep.
Hominy is nixtamalized, or it's and so you can't make a tortilla with cornmeal, right? Because cornmeal isn't sticky. But once you've boiled it in an alkaline solution, it changes the texture of the starch to make it sticky so that it'll hold together and you make a tortilla out of it. That also releases the B vitamins. But I was just curious. It's just interesting when you say that charcoal has this anti-inflammatory effect, because of course that is the native, bread of North America, at least, as it were, is this, corn soaked in literal wood ashes.
Yep.
Very interesting.
Yeah, and I guess I guess when you add, so it contains what, potassium hydroxide, the wood ash, right? It contains some kind of a base, which I think reacts with the starch and basically puts it in a semi-digested state. At least breaks down the complex particles so that they don't get, even if they get absorbed into the bloodstream, the immune system interprets it as a sugar and not as a complex particle like an endotoxin, because the endotoxin is lipopolysaccharide. Right. So it's a type of lipid and sugar combined. So as long as you can break it down further, then it's not going to trigger an immune reaction. That's probably the main benefit of and I think Dr. Pete has recommended hominy to several people.
Okay.
Who had trouble digesting other foods.
Even that, it's like a canned stuff, right? Is that what people eat? Yeah.
You get it again. I'll get just while we're doing it, I'll give people the recipe. It's very easy. So you take I don't know, I do it in a pot on the stove. I have a fireplace that I burn hardwood in, and it's hardwood I collect from my property. So I have clean ash, but you basically just take a couple of cups of ash, you put it into your 8-quart pot, bring it to a boil, and then you add just whole kernel corn. And it is shocking how difficult it is to find whole kernel corn. Like, you cannot drive around the countryside without driving past endless cornfields yet, right? If you want to just buy some whole kernel corn, they have it. I guess you can buy stuff for the birds at Agway or something. I happen to have, there's an organic mill near me that I can buy a 50-pound sack of whole kernel corn, which they use to make pig feed out of or whatever. But anyway, so you take a little bit of that and you add it to this water with the wood ash. You bring it to a simmer for about 5 minutes. You put the lid on it and you just let it slowly cool in the hot liquid. So you leave it on there for like 8 hours after it's simmered for like 5 or 10 minutes. And then what happens is the skins come off the corn. And so you do this thing, they call it, the word in Spanish is like refrigerar, which it just means rub. So you rub it, and that makes the kernels or the skins come off. Also, typically the germs come out. And interestingly, the germ is where most of the vegetable oil is. So most of the PUFA is in the germ. So after you rub it, and then what happens is you kind of rub it, and then you pour it through a sieve, you run water over it, just anything to kind of like, get rid of all the skins and the germs and the ash. And that leaves you with just the hominy. And that is, and you've removed, like I say, I guess it's pre-digested, and you've removed the PUFA.
The PUFA, yeah.
And it also tastes delicious, and it smells great, and it's a fun, easy thing. And obviously that's what people did for 10,000
years. It reminds me of
a
Mexican restaurant I like to go to here in Florida. They have the menudo, where the beef tripe and everything, and they've got hoof in it. And so, that's the ultimate Pete meal, huh? Because you've got they'll bring out a cup of this hominy with it. You pour it in there, and then you got your corn tortillas. That's the ultimate peak meal, huh?
Yeah, collagen, good carbs.
Yeah.
Easily digestible, non-endotoxin, low PUFA.
Well, Brad, speaking of that gelatin, Brad, your low PUFA pork, you're gonna have to give him some of that gelatin for your low PUFA.
Are you feeding the pigs like saturated fat or like butter, or how you making?
So we're feeding them basically we're giving them like a purified starch. So there's no what it is. We're actually feeding them a lot of pea flour, which comes from they grow field peas, and then they go to the factory in the Midwest, and they want the protein isolate from the peas. Right. And that leaves us with basically a pure starch that doesn't have any PUFA left in it. And that's the base of the diet that we use to fatten the pigs. Although I've thought about adding some saturated fat to it, but I'm not totally sure what the outcome of that would be. Like, I think you can get beef tallow, which would be an interesting to see how that affects the final fat composition of the meat. But I haven't done that experiment yet to figure out quite what the effect of that would be. But it's something I want to try. It'd be very interesting.
There is a pork farmer who emailed me maybe about a year ago. He wanted me to post, because I post on the Repeat forum. He wanted me to post about his farm, and he apparently is feeding his pigs mostly beef tallow and butter and basically ruminant fats, right? And he sent some pork for analysis, and he's keeping a control group of pigs as well to a farm, to a lab, and basically the results came back really good. He sent some of the pork to Pete, and you can tell because when you cook this pork, even before it gets cold, basically the water, like the stock from it becomes immediately hard, which means there's a lot of saturated fat in it.
Right.
While with the commercial pork, even the organic one that I get from Whole Foods, I've never gotten, even when I put it in the refrigerator, it just stays liquid.
So one of the things that I like to talk about, being a kind of veteran of the livestock industry, is that one of the problems, especially common with organic pork, is that, everybody thinks that they need to put soybean meal into the pig rations for the protein, which is not at all true. Yeah. But that's what everyone thinks. And so that's what they do. And so, soybean meal is typically, the commercial stuff is sent to a factory, and they grind it up, and they soak it in like hexane, and that gets the fat out of it, and then they dry it, and then that's what we make the pig feed out of. But the organic mills don't do that. They don't have hexane extraction locally and at small scale. And so what the organic feed mills will do is they'll just roast the soybeans. You can't feed raw soybeans to pigs because they can't digest the protein. And so they just roast whole soybeans and grind them up and put them in the pig feed. So if anything, the organic pig fat, unfortunately, might be higher in polyunsaturated fats. They might be the highest source of PUFA in pork is the organic pork, unfortunately. And I say that sadly because I'm friends with the owners of the local organic feed mill, and I don't want to say bad things about my friends, but that's the reality of it, right?
That's how it turns out, right? Because it has the most PUFA. Yeah.
Right. And so what you can do, and I published a study on this recently that was anyway, but you can actually just take whole ground barley and supplement it with the amino acid lysine and feed it to pigs over 100 pounds. And that's all the protein that they need. They don't need more protein than that. A pig grows fine on 12% protein as long as they have enough lysine. And so, we have this whole industry around growing all of these soybeans that we literally don't need. And there's a whole body of research out there on low protein pig feeding. It's like scientifically we know that we don't need all these soybeans to make pork, but, everyone can't.
That's what Congress subsidizes. It's just like the grants process.
Right.
For the next 20 years, money has been allocated for something. We're not going to stop it now, even though we know it's wrong.
And if you talk to, livestock nutritionists are literally the least creative people I have ever met on the planet. Like, I can't believe it. They will not. It's like, no, pig feed is corn and soybeans. That's what it is. Here's how you make it, period. Like, They're not going to talk about anything else. You know, they don't care. They just want it to be corn and soybeans. That's what they know. They went to Ohio State, and Ohio State told them corn and soy. That's how you feed pigs. And that's as far as that conversation will go. And then of course, when you go to the feed mills, they call those people that went to Ohio State, and they're like, well, yeah, no, pig feed, it's corn and soybeans. I have a funny, and just real quick, I know we're getting late, funny anecdote on this. So I in my, early days of raising the pigs and much more naive days, I called a local mill, and I was looking for this grain called triticale, which is a cross between wheat and rye. And it grows well. It's fairly high in protein. It's fairly low in PUFA, and I wanted to feed the pigs that. And a lot of people use triticale as like a smother crop or a cover crop. And so, some places sell triticale seed, and farmers will plant it as a cover or whatever. And so I called the mill and I said, hey, I'm looking for a local feed mill that will make me pig feed that's made out of the grain triticale. And she said to me, son, we sell feed, not seed. And I explained to her, I said, yeah, no, I understand this is a feed mill. And what I'm saying is I want to make a feed for my pigs, which is based on the grain triticale, that we could buy from a local farmer that grows this triticale. You buy it, you grind it into a feed and feed that to my pigs. And she just said louder and more forcefully, feed, not seed. It's like, the livestock industry, it's incomprehensible to them that you could feed a pig something other
than corn and soy, right?
It's just that triticale is not feed. It's not feed for pigs. And you're like, well, here's 5 studies showing that pigs grow really well, and they don't care. They don't care.
Sounds like the livestock industry is a lot like the medical establishment for humans too.
I was just about to give an example. There's something they call it the Ringer's lactate solution. It's basically it's a lactate in solution. They give it to people as a resuscitation thing in hospitals, right? Or somebody's coming in with already reductive stress, and already they have a lactic acidemia. Why on earth would you give them an additional lactate in this? And then these people collapse, right? And I've had this discussion with doctors who say, no, it's scripture. In medical school, they tell you give Ringer's lactate, and I don't care what this person has. So hold on a second. This person comes over already following with lactate, and you even give them more, and then you wonder why they collapse? Well, what would you do? I don't know, like give something that gets rid of the lactate. Oh no, that's different department. That's what we do. So yeah, I think this group think, and I think these people are institutionally damaged in a sense, that you cannot get a certain thoughts across their minds. To them, it's sacrilege. You just don't talk about these topics.
Well, right. I got to run, but I really appreciate you guys. This has been a fun discussion, and I hope that we can do some more down the road. Okay.
Awesome. Same. I'm always available. Thank you.
Georgi Dinkov's website is haidut.me. That's HAIDUT.me, correct?
That's correct. Yeah. And that thing feeds into Twitter, which is the same thing, twitter.com/haidut. If you want to keep up with, you get a more timely update on when I post new articles.
Nice. And Brad Marshall, you're at fireinabottle.net, correct?
Fireinabottle.net, and also on Twitter at fire_bottle.
Very good, guys. Thank you.